Can everybody see my screen?
Yeah, it looks good.
Okay, let me grab my pointer. Okay. Yeah, so I'm Amanda Burke. I am the Senior Field Application Scientist from Milton Biotech supporting our imaging portfolio. And I'm going to talk today about organic clearing, specifically the DISCO methods. Um, so we're going to talk about why, what, and how, everything tissue clearing related to iDISCO. And then, like I said, we have plenty of time at the end for questions and answers, but also feel free to stop me if you need any clarification. Let me move this so I can see. All right. Well, that doesn't work. There. Okay. So why do we clear tissues? So with 2D immunofluorescence, everybody knows that this is kind of like um, the most common practice, if you will, in imaging. But the data obtained represents the three-dimensional nature of biology in a very limited manner. So in comparison, renderings of a whole intact organ, oops, is the video playing?
I don't see a video, but I do see a really pretty heart. Is it a heart?
Oh, why
Yeah.
isn't my videos playing?
Or is it a brain? I don't know. It's okay. It's all right. Let's take
Okay,
a moment and think about this.
hopefully all of my videos aren't disabled. That's so strange.
Oh, you know why? Zoom.
Oh, really?
Yeah, sometimes the nature of Zoom. So I'm sorry, but I will say you have really pretty pictures, and I know it's much cooler if you can see them
That's too bad.
three-dimensionally. Um, and maybe we can express that to our users. Seeing things three-dimensionally is meant to be seen in video. So
Yeah.
that's
Well,
okay.
I apologize for that. It's not going to have the powerful, you know,
Wow factor. That's
yeah,
okay. It's
exactly, that I
Zoom.
hope
That's the
for.
nature of Zoom. Yeah, that's
Yeah.
sorry.
Yeah, but basically both of these samples were stained with the same antibody for CD31 vasculature. So it's clear that the 2D image on the left cannot capture the complexity that you would see in this 3D image, and the video really just drives that home.
Yeah. Mhm.
So if your goal is to capture an accurate portrayal of the spatial complexity of an entire organ, we need to apply 3D volumetric imaging techniques. But you may wonder, how do you achieve the most optimal results with 3D imaging? The intact samples are generally much larger than 2D physical sections. How are we able to see deep into the tissue? And you may have already guessed, that's through tissue clearing. Oh, this one's playing.
Okay.
Can you see this? Oh, good.
It's
Okay.
beautiful. It's really beautiful.
Okay, so let's kick off this tissue clearing concept with some very basic kitchen science to demonstrate why tissues are inherently opaque. So here you can see I am shining a laser pointer through two glasses. One contains just distilled water, and the other I have added a couple of drops of milk. So essentially, I've added a mixture of molecules composed of mainly proteins and lipids to the water. And by doing so, you can clearly see that the light passing through the glass that contains milk, the light is behaving much differently. We can actually see the light in the solution. But why does this happen? So in a given tissue sample, light passage is highly impaired due to things like reflection, refraction, scattering, and absorption as it encounters these different materials within it. As I alluded to in the kitchen experiment, these materials in a biological sample are largely comprised of water, proteins, and lipids. And each of these molecules have a very different property that affects the behavior of light, or they're defined by the refractive index. I also refer to this as the RI, just not to confuse you. Um, so the RI serves to quantify these properties, where the assigned numerical value represents the speed at which light travels through that given molecule. So to give you some perspective on the range of refractive index, or the RI, in a biological sample, water has a refractive index of 1.3, proteins around 1.5, and lipids 1.4. So this number is an expression of how much slower light moves through the substance compared to that in a vacuum. The higher the number, the slower the light travels through that material. So the goal of tissue clearing is to render the tissue transparent. I think everyone can kind of grasp that concept, clearing, transparency. But what does that really mean? It's essentially the result of homogenizing or matching the refractive index of the entire sample. So tissue clearing is going to utilize different chemical concoctions to homogenize the RIs of tissue through the removal, replacement, and modification of the tissue's composition. And you get a result like this in the end, maybe even clearer. Okay. So when it comes to tissue clearing, there are so many different protocols. That's why I'm here to help guide you. But ultimately, there are three major classes, and I just want to make you aware. There's hydrophobic clearing, hydrophilic clearing, and hydrogel-based clearing. But for the sake of this talk and for all of your applications, I'm going to really hone it in on hydrophobic or organic clearing. And that is because it can be applied to a variety of different tissues. It's really robust. It's really quick. Just to kind of give full disclosure, with this method, there is a little bit of tissue shrinkage, but as we get into the nitty-gritty details, I'll show you how the protocol has been adapted to try and reduce this. And then there are going to be some toxic substances in the actual clearing application itself. But I will talk about an alternative imaging solution. So at least during your imaging sessions in an open environment, this will be done in non-toxic circumstances. So for organic clearing, there are essentially three major steps. So that's dehydration, delipidization, and RI matching with an organic solvent. So it's very simple, and this can be accomplished in a couple of days, as you can kind of see the timeline as it goes here. So within this class of tissue clearing, one of the most well-known organic methods is 3-DISCO, which is essentially the building block of all these variations of discos. Like I said, I'm going to be talking about iDISCO, but there's B-DISCO, there's F-DISCO, U-DISCO. So this is the essential building block where all of those protocols stem from. So with 3-DISCO, dehydration of the tissue is accomplished with alcohol, either methanol or THF. THF is tetrahydrofuran. I'm not a chemist. I don't know if I'm pronouncing that correctly. The delipidization is done with DCM or dichloromethane. And then the final clearing step is done in DBE, which is dibenzyl ether. And all of these times, as you can see, the dehydration is pretty spot on. But when it comes to the clearing portion, there's a big range in time. And this is really just dependent on the size of your sample. This will be a common theme throughout my talk, because really a lot in terms of the timeline of clearing and staining will depend on your sample size.
Can I ask a question?
Please.
As we're talking about these chemicals, I love to ask, how nasty are they?
Yeah.
Are we talking chemical fume hood? Are we talking like,
yes.
You know, like how scary?
I would. I mean, it's not a lot of hands-on time, and I'll kind of get to that too when I go through the workflow. So I would absolutely recommend a chemical hood, especially for steps with the dichloromethane. That's probably the nastiest one. DBE, it is a toxic chemical. It's not as bad as DCM. So I kind of have a tip a little bit later. Maybe I'm jumping ahead, but
no,
yes, I would.
Whenever you
yeah,
want.
I would definitely work under a hood or make sure that you have a chemical hood around for the clearing part.
Okay.
Yeah. So for the 3-DISCO method, which is really the back end of iDISCO, I already have some tips. So first of all, it starts with a good perfusion fixation. If you can perfuse your tissues, the results are much better because it dries out all that blood. Blood is highly autofluorescent, and then having that really nice fixation from a perfusion that you can't always get with drop fixation. But don't overfix your samples. So overfixation increases tissue autofluorescence. If you're not moving into clearing right away, please don't store your samples in PFA or formalin for a prolonged amount of time. Put them in PBS with maybe some azide. When you're going through this process of fixation, dehydration, all of these steps really, fill the vessels completely. You don't want to leave any air. This can cause oxidation to your sample, and then you'll start noticing that the transparency is not as nice. If a sample is still opaque after you have it in DBE, let's say you're like, oh gosh, I'm just going to leave it overnight, it's still quite opaque. This usually means that it wasn't dehydrated all the way. So there's still some water molecules left in the sample itself. So I do see this a lot when people are reusing ethanol or methanol. Let's say it's an older bottle, a
Yeah.
lot of contamination of just environmental water molecules gets in there. So I always say use a fresh bottle of alcohol. Um, if you want to use something that's been open for some time, you can use molecular sieves to really pull out the water that may have accumulated in there, but really make sure that you're starting with um, fresh or uh, water-free alcohols. And then if the issue still persists, you may want to increase your 100% methanol step overnight. So really driving all the water out of there.
If somebody reaches that point where they do the RI clearing, it's not that great, can they bring their sample back into fresh alcohol? Like, can you go back in a
You can, you
okay.
can. I would try to avoid that. The results aren't always so great, but
Okay.
you can. But you would do it in a gradient manner. You wouldn't just throw it back 100%. So you would kind of cut the solution with your RI.
If so basically, if you need to go back in time, you need to go all the way back to that first dehydration step, run it through those again,
Yes,
and then go to the end. But the better thing is that you have good alcohol and you make it properly the first time.
yes,
Okay.
yeah. So this is a really common occurrence because people don't really think about ethanol going bad or methanol going bad, but it does accumulate water molecules as it's opened each time. So using fresh is always best.
Okay.
So 3-disco may work fine in some cases where maybe you have a really robust fluorescently tagged viral vector, so a lot of high expression, or if you just need to look at autofluorescence. This allows you to basically extract the organ and move straight into your clearing protocol, so 3-disco. But in most applications, immunolabeling is necessary to either visualize or enhance a fluorescent signal. So this requires some pre-treatment prior to clearing, and this is where I introduce iDISCO to you, and this is where that I comes in for immunolabeling. iDISCO, as I already mentioned, was built on top of 3-disco. So 3-disco is the back end. iDISCO incorporates some of those pretreatments and the antibody labeling of your samples. It was developed by Nicholas Renier. And 3-disco, I forgot to mention, was developed by Ali Irtuk. So both of them, if you want to stay informed on the disco methods, they're really the experts.
I have a question real quick from a user, from Martin. He says that DCM melts plastics. What do you use for that instead of DCM? What do you use as a container?
I will get that. I will
Okay,
get to that. I have a slide on all of that. And I will provide these slides to you as well, April, to distribute.
thank you.
So when it comes to iDISCO, here's a very simple breakdown of the workflow. So you may have noticed that the total time has increased drastically from 2 to 5 weeks. And that's because the immunostaining does take quite some time, and this is dependent on your sample size. So if you have a smaller sample, it can be accomplished in 1 week. If it's larger, 4 weeks. This also depends on your antibody system. If you're using a primary and a secondary, this doubles your time of immunolabeling. So that's really where that time comes in. But this doesn't necessarily translate into a lot of more hands-on time. You're still just essentially taking your sample and moving it into different solutions. So the payoff for the time is that you will have a much nicer signal than any endogenous fluorescent protein. The clearing protocol, no matter how you adapt the protocol, there will always be a loss in fluorescence of those endogenous fluorescent proteins. So the payoff is really that you get really nice signal and pretty images in the end. And if anybody was wondering, the DISCOs, it's a funny name, but it actually stands for 3-dimensional imaging of solvent cleared organs. And then the I is for immunolabeling. So since this talk is focused on iDISCO, I'm going to give a little bit more information on the chemical components and the workflow. So first, we're going to pre-treat the fixed samples in methanol to dehydrate the sample and already begin to disrupt that extracellular matrix for permeabilization. This is followed by an overnight bleaching step with hydrogen peroxide. This really reduces autofluorescence of the tissue. And then a mixture of DCM and methanol. This is already going to remove some of those lipids and really help with antibody diffusion. For the immunolabeling, an aqueous environment is required, so we're now going to rehydrate the sample with decreasing concentrations of methanol. The sample is then incubated in a permeabilization solution that contains DMSO and glycine, followed by a blocking step in serum. If you use antibodies that are recombinant antibodies, like the ones Milteney offers, you can skip this step, save some time. If you have any questions about those antibodies, feel free to ask. And then, where am I at? So we're going to block, and then we're going to label with our antibodies, with heparin, DMSO, and then again with that donkey serum if you're not using recombinant antibodies. So you may notice that the iDISCO, I added a little plus, and that is because the protocol has been updated with some enhancements. And one of those was that they found glycine and heparin prevent non-specific binding. And then finally, for that clearing part, which is essentially the 3-DISCO portion of the protocol, we're going to dehydrate the sample again with methanol, another delipidization step with DCM to really drive out all of those lipid molecules. These lipids are really responsible for a lot of the light scattering that you may see. So it's important to include that step at the end. And then finally, you're going to clear in DBE. Some people do like to clear an ECI to avoid DBE, but if you are doing this entire protocol, like we mentioned earlier, DCM is much nastier than DBE. So if you're already working under a hood, I always recommend to clear in DBE. Your samples will clear faster. It is a much more robust clearing. The ECI sometimes leaves like this yellowish tint to the samples. So if you're already going through this process and working under a hood, clear in DBE, and then after the sample is cleared, transfer to ECI for your imaging session.
I just want to jump in here and say on the
Yes.
ECI that we are doing all of our organic cleared samples are going to be imaged in ECI. So everybody is ending up in ECI at the end, please.
Yes.
Okay.
So the clearing aspect of tissues is relatively simple. I hope that I've conveyed that, right? You just bring it through some dehydration, you put it in RI matching solution, boom, you're done. It really is the immunolabeling portion that requires a little bit more consideration. And I wanted to devote a couple of slides here because this is where people really struggle to optimize their sample. So I want to give you some tips here. First of all, antibody compatibility. Most antibodies are completely compatible with this protocol, no problem, they can find their antigen. But there have been a handful of antibodies that have been tested that this methanol treatment prevents them from recognizing their antigen. So it affects the tertiary structure of the protein. So I always recommend if you're using an antibody currently and you know that it works nice in your tissue sections and you want to bring it over to 3D and tissue clearing, make sure that you test it for this methanol compatibility. So do a pre-treatment on sections. Sections are much smaller, cost effective. You won't be using as much antibody and it's much faster that you'll get your results and a confirmation that your antibody is working. So that is something that you want to do right out the gate if you're working with an antibody that you're not using methanol with. But if you can, try and find antibodies that have already been 3D validated. And what do I mean by 3D validated? So these are antibodies that have already been screened for functionality, optimized for tissue penetration, and titrated with a recommended working dilution. So someone else has already done all of that work for you. But if you have no choice, but to use an antibody that has not been optimized for 3D intact tissues, I do have some pointers based on a lot of feedback from my users. So one of the biggest challenges is what you see here on the left. It's this halo effect around the tissue. So it's really hard to diagnose whether this is an antibody concentration issue, whether this is an incubation issue, and it takes a lot of effort to get something like what you see on the right here. So these are my tips for the order of events or how you should tackle this. First, I always tell people probably the easiest thing is to incubate your sample longer with your antibodies. So increase, if you're using a primary, secondary, increase both of those times. And then usually that helps quite a bit. But if you don't see any improvement, then I say increase or modify your permeabilization step. So this would be in conjunction with the antibody incubation time. Modification, if you're really struggling here and you know that it's a permeabilization issue, um, there are some different variations to the iDISCO protocol that I can help you with. For example, in human tissues that have been overfixed, there is a detergent called CHAPS that really helps break into those samples and get those antibodies through. So there are some modifications that we can use depending on your tissue type. If you still don't see any improvement here, actually, people want to increase their antibodies, but this is more, it's usually more of an issue with the antibody concentration being too high. So I would suggest instead of increasing your antibodies to reduce them. A lot of times with these denser tissues, the antibodies that can't penetrate, they kind of bind to themselves and do this non-specific net that you'll see on the outside. So sometimes reducing the antibody really helps to give them enough room to spread out and enter the tissue. In conjunction with the reduced antibody concentration, you may notice that you're not getting as much signal as you would, expect to see. So in conjunction with reducing the antibody, I would also recommend performing multiple rounds of antibody labeling. So allowing more and more antibody to enter, but giving them enough space to not form this net here. If you still don't see any improvement, lastly, I'd recommend a titration. I don't like to say this from the beginning. Some people may just want to jump to doing an antibody titration, but with 3D samples, and if you're working with a large sample, this is a lot of antibody. So I try to avoid this and do it in the most cost-effective way of tackling this issue, but in the end, it may be that you'll have to do a titration. Another consideration is if it's available, try to get an antibody that's directly conjugated to the fluorochrome. As you can see here in this timeline, using the primary secondary system, it will double your protocol because I would never recommend to incubate your sample any less time than your primary. So however long you're incubating your sample with your primary antibody, you are going to do that with your secondary. So using an antibody that's already conjugated to a fluorochrome will drastically decrease your time frame here. Miltenyi does have a portfolio. If you are interested in understanding how these antibodies are validated in terms of specificity and things like that, but also for 3D, I'm happy to answer those questions. I think I have some slides at the end to bring those up if you want to know more about those antibodies. We also found with our antibodies that we get a better signal to background ratio. So this is some indirect labeling here, and then this is with our antibody with that fluorochrome. So our antibodies are really small. They are conjugated to a very small, very photostable dye, very similar to the Lexaflors. So basically bringing you through these tips for iDISCO, try to find antibodies that have already been validated, whether that's a vendor like Miltenyi selling these 3D antibodies, or you can refer to the iDISCO info page that I distributed to April. They have a list of antibodies, working solution, incubation times, and then go to the publications as well. So these antibodies have already been validated to be compatible with the methanol pretreatment that's necessary for the iDISCO. They already give you nice working dilutions and timing. So this already helps you a lot to get your results faster. If available, use a primary antibody directly conjugated to a small photostable dye, again, speeding up this process. And then in terms of light sheet, the redder the better. So try and stay away from 405. You get much better signal to noise and the more red portion of the spectrum.
I always jump in on this and remind people that there are nuclear stains in every wavelength of light. You don't have to use DAPI if you want to be able to see your nuclei. You can use something that's farther read out like DRAC5 or TOPO3.
So I do want to make a comment. DRAC5 was tested in the iDISCO paper, and it is not compatible.
JK?
However, TOPRO is, and most other nuclear DNA stains are. So you still have plenty to choose from. But
Okay.
DRAC5 is the exception.
Okay, sorry.
Yes. And then again, the tips for a 3-DISCO. So along with those antibody tips for the iDISCO, the back end, which is essentially the 3-DISCO protocol, they all apply here as well, right? So if you can perfuse, the dehydration steps, make sure that your vessels are filled to prevent oxidation, and then make sure that you have complete dehydration of your sample before bringing it into that final DBE step. Does anybody have any questions about the workflow?
I think I keep asking the same question again and again, and I'm really sorry. So I asked a question previously. If it's not cleared, very nice. Can you go back to the dehydration steps, which you answered? And then if you get to the very end and you get this ring where you see that your antibodies are not going in, first off, when do you check for that ring? Do you have to check at the very end once everything is cleared and you put it onto the
Yes.
menu? Okay, so if you see the ring of fire, let's call it the ring of fire. Can you go back in time all the way to the eye of iDISCO, or do you have to have a new sample?
So I do have a couple of users that were successful going back. But the issue is the antibodies are still going to be there on the periphery. So while you may get better penetration, if you go back, you'll still have all this non-specific on the edges. If that doesn't bother you so much and you're really just concerned with the deeper structures, it may be okay. But usually people go all the way from the beginning. That's why the optimization with 3D can be painful. That's why I give you these tips to smartly select antibodies and don't try and reinvent the wheel, because you do have to take it through this whole process, which takes a lot of time and resources because you're using a lot more antibody. So really try and take some time to do your homework or refer to me. I'll try and help you out because optimizing an antibody can be very time consuming.
And maybe the other point to this is don't be using super precious samples from patients out the gate. Maybe
That's right.
I think that's like just samples that you have laying around, not laying around per se, but like something that's not super precious, and understand, like, let's say you say this whole entire protocol can take weeks for the antibody stain, everything to go through. It could take a little while to optimize that process. So first off, don't try to cut corners. Try to do it properly out from the get-go. Second off, don't necessarily use your most precious samples to begin with. And third off, don't rush things like, we get sometimes people 2 days before grants due. That's not a good time. Okay.
No. So, I mean, I'm kind of jumping ahead, but there are lots of resources out there. So you bring up the grant. If you email me or reach out to me, there's typically some type of publication for this method, this tissue clearing method, that spans, all kinds of applications. So if you're applying for a grant and it's kind of something that you need quickly, you can usually refer to these publications. So I can help you acquire literature. Okay, so just some practical considerations about iDISCO. People like it because it's fast, it's inexpensive. Clearing takes hours, like we talked about. The immunolabeling is really the time suck from this method. The storage of tissues. April jumped in and she would like you to store it in ECI. I still recommend that you clear with DBE, but storage should be done in ECI. Make sure you're filling up those tubes to the top. Oxidation really occurs during this storage time. So it's after the sample has been stored for weeks, years. is when you notice that oxidation really kicking in. So make sure that you're filling your tubes completely.
What temperature are they storing it at in ECI, and is it dark?
So you bring up a great point about temperature. Long-term storage, I would store it in the refrigerator. If you notice that your samples kind of have like this white precipitate on them, that's something um, that's something specific to ECI. So if you put it out at room temperature, this will go away. Don't worry about it. I've had a couple people contact me about, their samples having this white stuff on it, and that's ECI, and it will go away once the temperature returns to room. With both DBE and ECI, you only want to use glass or polypropylene vessels. ECI is non-toxic, but it still will melt plastics. So a lot of people aren't aware and will store their samples in ECI. And I've had this for actually a demo. Somebody sent me their sample in ECI, and by the time it got to me, the entire vessel was eaten up by the ECI. So there was no demo. And then if you go with the Alexa dyes for your secondary, or if you can find one that's directly conjugated, and Miltini Vio dyes are very similar. These are highly stable for over a year. So you can go back and re-image those samples. Recommended imaging solutions, we already said ECI because it's non-toxic. It has a nice smell. I like it. It gets on some people's nerves, but if it does, you can always get a fume arm to get rid of that. And then whenever you are handling your samples before it goes in the scope, same thing. The ECI will melt the plastic, so you're going to want to use glass or stainless steel labware.
I want to
Just
Can I jump on this as well? I just want to say, I know a lot of folks have things hanging out in the lab that they constantly use for stuff like that. Please, please, please validate that it's polypropylene. So
yes.
like I would hate for somebody to spend so long going through the entire procedure and then just pull out a tube, slap their sample in the tube, and that very silly mistake to ruin everything for you. That would be a disaster. So please make sure that you know that this is polypropylene that you're working with. Okay.
Yes. And to that point as well, just from personal experience, ECI will also disrupt the finish on lab benchtops. So it is a cosmetic thing, but what I like to tell people to do is if they're working on their lab bench or even in the fume hood, use these, I call them blue diapers, but these lab pads,
Yep.
So that you're not putting the ECI bottle directly on top of the counter, because it will mess up the finish and leave these ugly looking stains that cannot be removed. And you'll find when you're working with this, especially ECI, it just tends to leak. I don't know how it does it, but it crawls up the side of the walls of the cuvette. It will run over. So just keep in mind that wherever you place this, it will mess up the finish of your lab benches. Some imaging considerations, like I said, there is some shrinking, and that's just, because of the dehydration step, as you may expect. There have been some modifications to the iDISCO Plus protocol to try and prevent that, specifically that DCM methanol pretreatment step in the beginning of the protocol helps prevent the shrinking from being so drastic. But the nice thing about these samples shrinking and not having that water is that they are easy to handle. So you'll notice that your samples get really rigid and hard, unlike the aqueous methods where you have like a gummy-like structure. So people tend to, after their sample is cleared, when they're trying to put it on the microscope, forceps really damage the sample. So these samples are going to be really easy to work with, and you'll see that during the training. Makes life much easier.
I want to say one more comment on this. I'm sorry, I'm adding a lot of comments.
Please.
So we kind of talked about this idea of using aqueous clearing for expansion microscopies. The idea of that is you blow it up on purpose so that you can see these smallly resolved structures kind of a little bit blown out, and you can get that resolution because of it. It's probably likely a little bit the opposite here. If you have some sort of shrinkage, you don't want to be looking at super duper duper tiny structures, because as it shrinks down, that's going to be reduced. Would you agree with that, Amanda?
Yeah, to a certain extent. I mean, I will say that they did test how shrinkage of the sample affects things like projections or connectivity patterns in a given tissue. And it doesn't seem that it really affected these factors. So when you're getting really small, like subcellular,
Yeah, that's what
um,
I'm worried about. Yeah.
it's kind of a double-edged sword because these clearing methods are really robust and will give you really nice transparency compared to some of the aqueous methods, but at the cost of shrinking the sample, like you said.
Yeah.
So if we're getting subcellular, we should have a conversation about what you really need to get out of that because it might be out of the limits of the scope as well, the microscope. So the nice thing about iDISCO over the past decade, a little bit longer. This field has been exploding. So as you know, light sheet is becoming recognized as a very powerful tool. People love it because they don't have to section anymore. They can get the full picture of their entire sample in this 3D spatial space. So there are lots of resources out there. This is one of my favorites. I recommend everybody kind of going through this. You can see how these methods evolved over time. Here are all those discos that I was talking to you about that all kind of stem from this 3 disco method. It has a nice table that provides different samples, both mouse and human, on all the different tissues that were cleared. So if you're looking for a protocol for a specific sample and you want to stay in this disco realm, it's a really nice overview and helps to guide you where you should go. And the author is Ali Ertug. So I mentioned him earlier. He was the one that developed 3 disco and really is the expert in this clearing method. There's also a dedicated page for iDISCO. I will say I don't think that they've updated it since 2000, maybe 19. So I don't know if this is something that they will continue to update, but it still holds lots of really good information. For instance, it will give you incubation time. So there's a whole list of samples that were tested here. So it gives you some idea of the volume of antibodies that you should be using, the time required for that antibody. And speaking of antibodies, it also lists all the antibodies that have been tested by this group. And it'll tell you the working solution that you should use for each. So really valuable information to, kind of save you from the headache of starting from scratch. And that's it for clearing. Just to reinforce, you can always contact me if you need help developing a workflow. We also have a technical support group that's available Monday through Friday, 24/7. You can give them a call. I will say they may not be so helpful with clearing, but they're really great with the microscope and helping you troubleshoot. And this one works too. So this was a brain that was cleared by iDISCO and stained for CD31 to look at the vasculature.
It's beautiful.
And any questions, discussion?
Also, I know we have recorded this. We will send this out if your folks didn't make this particular talk or if they have any questions after the fact. Thank you, Amanda, for helping us walk through this protocol. I don't think that the protocol at least seems incredibly complex per se, but I think one of the things, bye Paka, you're welcome. One of the things that seems to be very apparent is do it right the first time. Take your time, have those fresh methanol solutions, have the right tubes, have everything done. It is significantly easier to walk through things correctly the first time versus trying to fix mistakes that have been rushed through these particular steps. So just take your time with your sample. Also reach out to us if you have any questions about experimental design or when you are ready to image, and we will answer those questions. I will send out the link from the Doodle link for people to sign up if they have sample in hand now for Miltenyi demo. And if they don't worry about it. We will be offering future dates in the future and having those conversations with all of our users one-on-one. So are there any other questions, comments, or concerns? No? Okay. right, I'm going to stop recording this and